We tend to focus on the big elements of life.
Yeah.
Carbon, oxygen, hydrogen, but kind of lurking in the background of the periodic table are these tiny trace elements.
Yeah.
And they wield an influence on our health that's nothing short of astonishing.
Mhm.
Today, we're zeroing in on one particularly fascinating group within these trace elements, the transition metals. If you've ever wanted to truly understand why these metals, needed in such, well, minuscule amounts, are absolutely critical for life,
Yeah.
then you've definitely come to the right place for a deep dive.
Exactly. And in this exploration, we're going to really dissect the vital roles played by essential transition metals. Think iron, zinc, copper, molybdenum, and others within the incredibly complex machinery of our bodies.
Yeah.
We'll be drawing directly from the detailed world of biochemistry to see how these micronutrients, required in what seems like, impossibly small quantities, drive a truly vast array of biological processes.
And when we say detailed biochemistry, we mean getting right down to the molecular level, looking at the actual blueprints of how these elements interact. Our mission today is basically to unpack the fundamental reasons why these metals are so essential, the ingenious ways they operate within us, and crucially, what the implications are when things go, well, off kilter. We're aiming for a really robust understanding, the kind professionals strive for, so let's plunge into the core principles.
Let's do it. So, what exactly are these essential transition metals we're talking about?
Well, simply put, they are a specific collection of metallic elements that our bodies absolutely must have to function correctly. The term essential here is key. We literally can't live without them.
Right.
And while they are metals, we need them in incredibly small amounts, which is why they fall into the category of micronutrients. The main players we'll be focusing on are iron, Fe, manganese, Mn, zinc, Xenon, cobalt, Co, copper, C, nickel, Nin, molybdenum, Mo, vanadium, V, and chromium, Cr. Micronutrients, it's almost humbling, isn't it, to realize how little our bodies actually need of these things to keep everything running. But as tiny as the quantities are, their impact is just immense, wouldn't you say?
Absolutely. Huge. Think of these transition metals as the unsung heroes at the heart of countless biological molecules. They are integral components of a huge variety of proteins and enzymes, those biological catalysts that dramatically speed up chemical reactions within us.
Workhorses.
Exactly. They are also indispensable in proteins involved in electron transport, which is fundamental to how we produce energy, and in oxygen transport proteins like hemoglobin in our blood and hemocyanin in some invertebrates.
Okay, so they're like the active centers, the key working parts, and a lot of these really important proteins. Could you give us some specific examples of how they actually function, at that molecular level?
Certainly. Let's take zinc, for instance. It plays a critical role in structures called zinc fingers. These are like molecular recognition modules found in many transcription factors, proteins that bind directly to our DNA and control which genes get turned on or off.
Wow.
The zinc ion acts like a structural anchor, basically, helping the protein fold into a specific finger-like shape that allows it to grab onto specific DNA sequences. Then we have iron-sulfur clusters, often just called FS clusters.
Okay.
These are found in a wide array of enzymes that are essential for processes like replicating our DNA when cells divide and repairing any damage that occurs to our genetic material. The iron and sulfur atoms within these clusters are directly involved in the enzyme's activity.
So zinc is literally helping to dictate our genes, and iron and sulfur are part of the machinery keeping our DNA intact. It's astonishing to think this is all happening thanks to elements we need in such trace amounts. But what happens if we don't get enough? What are the consequences?
Yeah, that's critical. Deficiencies in these essential transition metals can have significant and sometimes quite severe health consequences. A lack of iron, for instance, can lead to pernicious anemia, a condition where the body just can't produce enough healthy red blood cells.
Right, I've heard of that.
Copper deficiency can result in Menkes disease, which is a rare genetic disorder that disrupts the body's ability to handle copper properly. And a shortage of molybdenum can cause sulfite oxidase deficiency, a really serious condition we'll definitely delve into a bit later. It just underscores how crucial it is to maintain adequate levels.
So clearly, not getting enough is bad news. But I also remember reading that too much of these metals can also be a problem. It sounds like a really delicate balancing act our bodies have to perform, doesn't it?
Precisely, a very delicate act. While these transition metals are essential in trace amounts, exceeding the body's carefully regulated levels can absolutely lead to toxicity. In fact, many of what we call heavy metals, which can include some of these essential transition metals if they're present in excessive quantities, are potentially carcinogenic.
Carcinogenic, wow.
Yeah, meaning they can increase the risk of cancer. Our bodies have evolved very sophisticated, very intricate mechanisms to control both how much we absorb and how we get rid of any excess to maintain that crucial equilibrium.
That brings us to a really interesting property of these metals, multivalency. What exactly does that term mean in this context, and why is it so important biologically?
Multivalency, in essence, refers to the remarkable ability of transition metals to exist in multiple oxidation states. Think of oxidation states as the number of electrons an atom has either gained or lost.
Okay, like different electrical charges.
Exactly. Many of these metals, like iron, copper, and manganese, can exist as ions with different charges. And this flexibility, this ability to readily switch between these valence states by either accepting or donating electrons, is absolutely fundamental to their role as electron carriers in what we call oxidation reduction reactions, or redox reactions for short.
Redox, right?
It's this dynamic give and take of electrons that fuels a vast number of essential biochemical processes within us.
So they're acting like tiny rechargeable batteries inside our cells, constantly taking on and releasing electrons to power all those reactions that keep us alive.
That's actually a very good analogy, a very apt way to put it. And this ability to participate in these electron transfer reactions isn't the only remarkable property these metals have. They also exhibit important Lewis acid properties.
Lewis acid. Okay, now, I remember learning about acids and bases way back, but Lewis acid might need a little unpacking for everyone.
Sure. When we typically think of acids, we often think of Bronsted-Lowry acids, substances that donate protons, right? Those positively charged hydrogen ions.
Yeah.
A Lewis acid, in contrast, is defined by its interaction with electrons, not protons. It's an aprotic species, doesn't donate protons. Instead, a Lewis acid is an electron pair acceptor.
Acceptor, okay.
They have vacant valence orbitals, sort of empty spaces in their outermost electron shell, that can non-covalently associate with or accept a lone pair of electrons from another molecule, which we then call a Lewis space. Many transition metal ions, like F2 plus, Zn2 plus 6, and Mn2 plus AC, act as very effective Lewis acids in biological systems.
Okay, can you give us some tangible examples of how this Lewis acidity actually plays out inside us? How does it help things along?
Certainly. Consider the crucial process of oxygen binding in our blood. When the iron ion, Fe2+, within hemoglobin and myoglobin binds to an oxygen molecule, it's essentially acting as a Lewis acid, accepting some electron density from the oxygen. Similarly, divalent zinc, Zn2+, or manganese, Mn2+, ions often play key roles at the active sites of hydrolytic enzymes, those enzymes that break down molecules using water. They function as Lewis acids by interacting with nearby water molecules, enhancing their nucleophilicity.
Nucleophilicity, meaning how eager it is to attack something.
Exactly. How eager it is to donate electrons and attack a positive center. By making the water molecule more reactive, these metal ions facilitate the hydrolysis reaction, making it much, much more efficient.
So they're not directly breaking bonds like a proton might, but they're sort of setting the stage, making it easier for water to do its job. That clarifies it. Now, we touched on the toxicity of heavy metals earlier. What actually defines a heavy metal and what makes them so dangerous?
Well, the term heavy metal doesn't have one single universally agreed upon definition, but it generally refers to metallic elements that have a relatively high density, typically greater than 5 grams per cubic centimeter, or maybe an atomic number greater than 20.
Okay.
Some well-known examples of toxic heavy metals include arsenic, antimony, lead, mercury, cadmium. Their toxicity in biological systems can arise through several key distinct mechanisms.
All right, let's delve into those mechanisms. How exactly do these heavy metals mess things up and cause harm?
One significant way is through displacement of essentialtations. Because some heavy metals can have a similar size and electrical charge to the essential metal ions our enzymes need, they can sometimes basically trick their way into the binding sites, effectively kicking out the metal that's supposed to be there.
Like an imposter.
Exactly. A classic example is when gallium, J3+, replaces iron, Fe3+, in vital enzymes like ribonucleotide reductase, essential for DNA synthesis, and superoxide dismutase, an important antioxidant enzyme. While gallium looks similar to iron, same charge, similar size, it lacks iron's crucial ability to cycle between oxidation states, its multivalency.
Right.
So when gallium takes iron's place, the enzymes just become inactive, useless, disrupting critical cellular processes.
That's a great analogy, like putting the wrong key in a lock. It might fit, but it won't turn. What's another way heavy metals cause problems?
Another really significant mechanism is the inactivation of enzymes by binding to sulfhydryl groups. Many enzymes have specific amino acid residues, particularly cysteine, in their active sites or on their surface. These cysteines contain sulfhydryl SH groups that are absolutely crucial for activity or structure.
Okay, the S-shaped groups.
Heavy metals have a very high affinity for these sulfur atoms and readily form strong chemical bonds with them. If this happens in the active site, it can directly block the enzyme's function. Even if it binds elsewhere on the surface, it can disrupt the protein's overall shape, making it lose its function.
So it messes up the shape or blocks the active part.
Precisely. We see this, for instance, with lead-inhibiting aminolevulinate synthase, an enzyme vital in making for hemoglobin. Similarly, arsenite and mercury can knock out the pyruvate dehydrogenase complex, key in energy production, by reacting with lipoic acid, which also has essential sulfur atoms.
So they're basically throwing a wrench in the works by physically blocking or distorting critical parts of these enzymes. And I also recall hearing something about heavy metals and generating harmful molecules. ROS.
Yes, that's the third major mechanism, the formation of reactive oxygen species, ROS. Heavy metals can act as catalysts in reactions that produce these highly reactive molecules. These ROS can then damage all sorts of vital things in our cell's DNA, the lipids in our cell membranes.
Damaging DNA and cell walls.
Exactly. Oxidative damage to DNA can lead to mutations, increasing cancer risk. Lipid peroxidation can mess up membrane integrity, affecting nerve impulses, nutrient transport. This is particularly bad for neurological and neuromuscular systems. And there's growing evidence linking chronic heavy metal exposure to tumors in animal studies.
It really paints a clear picture of why heavy metals are such a concern. Not just sitting there, they actively interfere with essential processes on multiple fronts. Now, we also mentioned that even the essential transition metals, the ones we need, can be toxic if we get too much. How does that work?
Right, it's all about balance. While absolutely vital in tiny amounts, exceeding the body's requirements for essential transition metals can indeed be harmful. Our bodies have evolved very strict control mechanisms for uptake and excretion to prevent this, like the hepcidin system for iron regulation.
Hepcidin.
But these systems can get overwhelmed if exposure happens through unusual routes, like inhaling high concentrations of metal dust, absorbing large amounts through the skin, or, accidentally or intentionally ingesting massive amounts.
And what are the telltale signs if someone has had an acute overdose of these metals, whether essential or just heavy metals?
Well, the typical symptoms of acute poisoning can include severe GI distress, intense abdominal pain, nausea, vomiting, diarrhea. You might also see neurological symptoms like confusion, muscle cramps, maybe numbness or tingling. The specifics often depend on the metal and the dose.
Sounds pretty nasty. Are there effective treatments available for this kind of poisoning?
Yes, fortunately, there are several strategies. A common one is using metal chelating agents. These are special molecules that bind very tightly to metal ions, forming a stable complex that the kidneys can then excrete more easily.
Chelating agents, right?
Diuretics, which increase urine production, can also help flush out excess metals. In severe cases, especially if kidney function is hit, hemodialysis filtering the blood externally might be needed.
It's sobering, isn't it? How these elements can be both essential and toxic. It brings us to how our bodies manage these reactive metals. You mentioned organometallic complexes. What exactly are those, and why are they so crucial?
That's a really critical point. The levels of free, unbound transition metal ions in our bodies are normally kept incredibly low, almost undetectable. The vast majority aren't just floating around. They are carefully sequestered, bound within these specialized structures called organometallic complexes. These are basically molecules where a metal ion is coordinated or bound, either directly to protein side chains, usually through oxygen, nitrogen, or sulfur atoms in amino acids like aspartate, glutamate, histone, cysteine, or indirectly via other organic molecules like porphyrin rings in heme or corrin rings in B12.
So it's like these metals are meticulously packaged and held in a very specific way. What are the key benefits of doing that? Why is complexation so important?
There are several really key advantages. Firstly, and maybe most importantly, it protects against unwanted oxidation. Free metal ions are reactive. They can react with oxygen. This uncontrolled oxidation can change their state, making them useless or even harmful.
Stops them rusting, basically.
Sort of, yeah. Secondly, binding the metals suppresses the formation of those damaging reactive oxygen species we talked about. Free metals can catalyze ROS production. Thirdly, complexation often advances the metal's solubility in water, making it easier to move around the cell.
Okay, protection, stops ROS, better solubility. What else?
Fourthly, it allows for very fine-tuned control of the metal's reactivity. The surrounding ligands, the bits bound to the metal, can precisely influence how it interacts with other molecules or participates in reactions. And finally, it lets you assemble multiple metal ions into a single functional unit for really complex reactions.
That makes perfect sense, like having specialized tools, each carefully designed and stored safely. You mentioned maintaining the correct oxidation state is critical. Why is that so vital?
The function of many metal-containing cofactors absolutely depends on the metal being in the right oxidation state. Take hemoglobin, the iron, must be in the Fe2 plus state to bind oxygen. If it oxidizes to F3 plus EI, you get methemoglobin, which can't carry oxygen.
Right.
The porphyrin ring and the surrounding histidine residues in the globin protein act like a protective shield, preventing this unwanted oxidation of F2 plus cells.
Like a little bodyguard for the iron atom. And you also said the surrounding ligands can fine-tune the metal's properties. How does that work?
Exactly. The precise identity and arrangement of ligands around a transition metal can significantly modulate its redox potential, its tendency to gain or lose electrons, and also its Lewis acid potency. This allows the metal to be optimized for very specific jobs.
So subtle changes make a big difference.
A huge difference. Think about cytochrome C and myoglobin again. Both have heme iron. But in myoglobin, the environment favors F2 plus for oxygen binding. In cytochrome C, the environment is designed to let the iron easily flip between F2 plus and F3 plus to shuttle electrons efficiently in the electron transport chain.
It's truly remarkable how changing the molecular neighborhood around one metal ion can dramatically alter its biological role. And you brought up superoxide dismutases, SODs. How do they illustrate this idea of adapting different metals for the same job?
SODs are a fantastic example. They all catalyze the exact same crucial reaction, detoxifying superoxide radicals, O2, into less harmful hydrogen peroxide and oxygen. But what's fascinating is there are 4 distinct families of SODs, and each uses a different transition metal at its active site.
Different metals for the same reaction.
Yep. We have iron SODs, FOCs, manganese SODs, MnSODs, nickel SODs, Ni SODs, and copper zinc SODs, Q xenon SODs. The specific protein structure around the metal in each family creates the ideal microenvironment for that particular metal to efficiently do the job. It's beautiful evolutionary adaptation.
So nature found multiple ways to solve the same problem, using different metals tailored to their protein homes. It's really elegant. And you also mentioned complexation allows organizing multiple metal ions together. What's the advantage there?
Now, bringing multiple metal ions close together in one enzyme, you can achieve catalytic feats impossible for a single metal ion alone. Take urease, found in plants and microbes. It hydrolyzes urea, critical for nitrogen metabolism. This needs the coordinated action of 2 nickel atoms.
2 nickels working together.
Yeah, one activates water, the other helps polarize the urea molecule, making it easier to break down. It's teamwork at the molecular level.
Like 2 specialized tools working in sync. What are some other examples of this multi-metal synergy?
Another key one is cytochrome oxidase, complex 4V of the electron transport chain, the final step in generating most of our energy. Its intricate contains 2 iron atoms and 2 copper atoms, all working together perfectly to collect 4 electrons and reduce oxygen to water.
Wow, 4 metals.
And then there's bacterial nitrogenase, essential for converting atmospheric nitrogen into ammonia, which life can use. It employs this incredible array of metal centers. An 8 iron 7 sulfur P cluster, a unique iron molybdenum cofactor, plus other iron sulfur clusters, all cooperating to pull off this really difficult nitrogen reduction.
These multi-metal centers sound like incredibly sophisticated molecular machines. Amazing how simple metal ions can be orchestrated like this. Now let's shift focus to the specific roles of some individual essential metals, starting with iron. You've mentioned it seems involved in a huge range of processes.
Iron is indeed incredibly versatile. As we've said, the heme-bound F2 plus in hemoglobin and myoglobin is crucial for oxygen transport and storage. And interestingly, some marine invertebrates use a protein called hemerythrin with a diiron penner for the same job.
So iron is fundamental for oxygen delivery across different life forms, but its roles go way beyond that, right?
Absolutely. Iron atoms in heme groups of cytochromes, B and C types, and in iron sulfur, Fe-S clusters, and Rieske iron centers, all parts of the mitochondrial electron transport chain are essential for transporting electrons. They do this by readily cycling between their +2 and +3 states, passing electrons along the chain to generate ATP.
Like a microscopic bucket brigade of electrons powering our cells. What about iron's role in actually catalyzing reactions in enzymes?
Iron is a key catalytic center in a vast number of redox enzymes. For instance, steroyl acyl carrier protein 99-desaturase, involved in making unsaturated fatty acids, and type 1 ribonucleotide reductase, crucial for DNA synthesis, both use diiron centers. Methane monoxygenase in bacteria uses a diiron center to oxidize methane. And the huge cytochrome P450 family, vital for detoxification and making steroid hormones, contains heme iron that forms a highly reactive iron oxo species. FeO3+, acting as a potent oxidant, cycling through multiple oxidation states, +2, +3, +4, +5, to reduce oxygen.
It really seems iron is right at the heart of so many important metabolic transformations. Are there roles for iron that don't involve redox, the electron transfer?
Yes, indeed. Some important non-redox roles, too. Consider the purple acid phosphatases. These are bimetallic enzymes, typically one iron paired with zinc, manganese, magnesium, or another iron. They hydrolyze phosphomonoesters.
Okay.
Myeloperoxidase, used by immune cells, uses its heme iron to make hypochlorous acid, HCl, a potent antimicrobial agent to kill pathogens. And intriguingly, many enzymes involved in DNA replication and repair helicases, polymerases, repair enzymes contain FES clusters. Their exact roles are still debated, but they might be damage detectors, redox modulators, or just structural stabilizers.
So even when not shuffling electrons, iron seems to play crucial structural or regulatory roles. Now let's move to manganese. What are its main functions? Where do we usually find it?
Compared to iron, humans have fewer manganese enzymes, and many are in the mitochondria, the cell's powerhouses. Key examples include isocitrate dehydrogenase in the TCA cycle, glutamate synthetase and arginase in nitrogen metabolism, pyruvate carboxylase, and PPCK in glucose synthesis, and mitochondrial SOD, MnSOD.
Mostly mitochondrial then.
Mostly, yeah. In most of these, the manganese is Mn2+, and generally thought to function primarily as a Lewis acid, like we discussed for zinc, helping stabilize transition states and facilitate catalysis.
So often stabilizing things or making reactions easier via its Lewis acid properties. Does manganese also do redox reactions?
Yes, in certain bacteria, manganese plays a more prominent redox role. Enzymes like bacterial MnSOD, Mn-ribonucleotide reductase, and Mn catalase use manganese ions cycling between +2 and +3 states to catalyze their reactions.
Okay, now let's turn to zinc. We've touched on zinc fingers and its Lewis acid role. What makes zinc stand out among these metals?
Zinc, Zn2+, is pretty unique because its valence electron shell is completely full. Because of this stable configuration, unlike many others, zinc doesn't readily adopt alternate oxidation states under physiological conditions.
Doesn't change its charge easily.
Right, which means it's not typically involved in direct electron transfer or redox reactions. This lack of redox activity also minimizes the risk of zinc generating harmful ROS, which is a potential issue with some other metals.
So sticking to +2 is its key feature. How does that stability help its biological roles?
This redox inertness makes Zn2+ exceptionally well-suited for structural roles in proteins. It can bind readily, helping stabilize the correct 3D shape, which is essential for function. It's estimated we have around 3,000 different zinc-containing metalloproteins, many being transcription factors or other DNRNA-binding protein, often using those zinc finger motifs.
We talked about zinc fingers. Just quickly recap how the zinc helps them bind DNA or RNA.
Sure. Zinc fingers are usually small loops of polypeptide chain. Their 3D structure is stabilized by a Zn2+ ion coordinating with conserved cysteine sulfur atoms and histidine nitrogen atoms residues. The zinc acts like a structural brace, holding the loop in that specific finger shape to make precise contacts with DNA or RNA sequences.
Okay.
The variations in the amino acids within the loop give the high site specificity. That's why zinc fingers are so interesting for potential genetic engineering and gene therapy designing tools to target specific DNA sequences.
Fascinating potential there. And beyond zinc figures and structure, where else is zinc crucial?
Zn2+ is an essential component in the active site of a vast number of metalloenzymes doing incredibly diverse jobs. Things like carboxypeptidase, A, protein digestion, carbonic anhydrase 2, CO2 transport, adenosine deaminase, purine metabolism, alkaline phosphatase, phosphate removal, phospholipase C signaling, cytosolic SOD antioxidant, alcohol dehydrogenase, alcohol metabolism.
Huge list.
Yeah, and even bacterial lactamases, which break down antibiotics like penicillin. In these enzymes, Zn2+ typically uses its Lewis acid properties, stabilizing negative charges, polarizing carbonyl groups, enhancing water's nucleophilicity, all boosting the enzyme's efficiency and specificity.
So its stable +2 charge makes it a reliable structural piece and a dependable Lewis acid catalyst without the redox complications. Okay, let's turn to cobalt. Its main role seems tied up with vitamin B12, right?
Absolutely correct. The primary, and as far as we know, the only biochemical function of dietary cobalt in humans is as the central component of 5-uro-deoxyethanosocobalamin, an active form of vitamin B12.
D12.
Within this cofactor, a CO3 plus ion sits in a corin ring system. It acts as a Lewis base here. It can donate an electron pair. This lets it bind and help transfer various one-carbon units like methyl, CH3, or methylene Nash's 2-DH groups in crucial enzymatic reactions.
What are the key human reactions that need this B12 cobalt complex?
In humans, B12, and therefore cobalt, is essential for two main enzymatic reactions. One is methionine synthesis, transferring a methyl group from 5-methyltetrahydrofolate to homocysteine to make the amino acid methionine, crucial for protein synthesis and more.
Okay, methionine synthesis.
The other is rearranging methylmalonyl-CoA to succinyl-CoA. This is a step in breaking down propionate, which comes from metabolizing certain amino acids and lipids. In this reaction, the CO3 plus briefly reduces to SOCO2 plus myelate, generating a reactive radical intermediate that helps the rearrangement. We cover B12 in more detail elsewhere, but those are the key roles here.
Okay, so cobalt, via B12, is vital for specific metabolic pathways involving single carbon transfers and breaking down certain molecules. Next up, copper. It's important, but I know copper imbalances can cause serious issues like Wilson's disease. What are its main physiological roles?
Copper is a component of about 30 different human metalloenzymes, showing its diverse functions. Key examples include cytochrome oxidase, electron transport, dopamine hydroxylase, neurotransmitter synthesis, tyrosinase, melanin production, cytosolic Q, ZN-SOD, antioxidant, and lysyl oxidase, collagen elastin structure.
A fair few important ones there.
Yeah, and dopamine hydroxylase and tyrosinase are interesting. They're catecholamine oxidases. They oxidize phenol rings in molecules like L-dopamine and tyrosine. Dopamine hydroxylase is key for making adrenaline. Tyrosinase is the first step in making melanin pigment. Both have a dicopper center where two copper atoms bind and activate oxygen, cycling between +2 and +1 states during catalysis.
So copper is involved in making hormones, neurotransmitters, and our body's pigments. What about its other functions?
So another interesting copper protein is hemocyanin. Unlike the oxidases, it transports oxygen in some invertebrates like mollusks and arthropods. It also uses a dicopper center.
Like hemoglobin, but with copper.
Exactly. And as we said, copper is crucial in cytosolic Q, ZN-SOD, working with zinc to detoxify superoxide. The copper cycles between Cu2 plus and C1 plus during the reaction. Then there's lysyl oxidase vital. It uses a single Cu2 plus ion to convert lysine amino groups in collagen and elastin into reactive aldehydes.
Aldehydes.
Yeah, these aldehydes then form crosslinks between collagen and elastin fibers, giving them their tensile strength and elasticity crucial for connective tissue integrity. Lysyl oxidase also has this unique modified amino acid in its active site, a quinone, that it makes itself from tyrosine.
Fascinating how copper participates in such diverse processes, from oxygen transport to the basic strength of our tissues. Now let's move to nickel. We hear less about its specific role in humans compared to others.
That's a fair point. While various nickel enzymes are known, especially in bacteria and archaea, the specific molecular need for nickel in humans and mammals isn't definitively established yet. We know nickel is in bacterial enzymes like hydrogenase, methyl coenzyme, M reductase, energy metabolism, acetyl-CoA synthase, carbon fixation, even a NiSOD.
And urease, you mentioned.
And urease, yes, where it helps break down urea. But in humans, while generally considered essential, its precise functions and deficiency effects are still largely unknown and being researched.
So clearer roles elsewhere, but still a bit of a puzzle in humans. What about molybdenum? I know it's critical for several important enzymes.
Molybdenum is indeed key. It's an essential component of a complex cofactor called molybdopterin. In animals, molybdopterin is required for enzymes like xanthine oxidase, aldehyde oxidase, and sulfite oxidase.
Okay.
Xanthine oxidase, which also has FAD and FES clusters, catalyzes the final steps in making uric acid from purines. The molybdenum cycles between +4, +5, and +6 states. Aldehyde oxidase, also with FAD and FS, has a broad range of substrates and likely plays a role in detoxifying foreign compounds, similar to cytochrome P450s.
And you mentioned sulfite oxidase earlier. What's its role and why is deficiency so severe?
Sulfite oxidase is in the mitochondrial intermembrane space. It's an iron and molybdenum enzyme that oxidizes sulfite, SO32, a toxic byproduct of sulfur molecule breakdown, to the much safer sulfate, SO42.
Detoxifying sulfite.
Exactly. Like xanthine oxidase, the molybdenum cycles between +6, +5, and +4, passing electrons ultimately to the electron transport chain via cytochrome C. Deficiency, usually from mutations in molybdopterin synthesis genes, MOCS1, MOCS2, GPHN, leads to a dangerous buildup of sulfite and related sulfur compounds in newborns.
And the consequences.
Extremely severe. Often significant physical deformities, profound brain damage, seizures, severe mental retardation. Tragically, it's often fatal in early childhood. It starkly shows how critical these trace elements and their enzymes are for basic metabolism and health.
That really underscores the impact these tiny components have. We're nearing the end with vanadium and chromium. Their roles seem particularly elusive, don't they?
That's accurate. Although vanadium is generally considered nutritionally essential, its precise role in humans is largely unknown, still under research. No definitive vanadium cofactor has been found in us. It exists in +4 and +5 states, binds plasma proteins like albumin and transferrin.
But it's used elsewhere.
Interestingly, it's a key component of the glue muscles used to stick to rocks. In lab studies, vanadate, similar to phosphate, inhibits some phosphatases, but the physiological significance in vivo is unclear.
And chromium.
Also enigmatic. Back in the 50s, a CR3+ containing glucose tolerance factor was implicated in glucose metabolism, but extensive research since hasn't consistently found a specific chromium biomolecule or related genetic disease to confirm its essential role.
But it's in supplements.
Despite the lack of robust evidence, yes, chromium 3 picolinate is widely used in supplements, often marketed for weight loss or blood sugar control. It highlights that there are still mysteries to unravel.
Definitely a reminder that we don't know everything yet. Now, before we wrap up, let's briefly touch on how our bodies actually absorb these metals from our diet and transport them. It can't be simple, right?
You're absolutely right. It's complex, highly regulated, and generally, the efficiency of intestinal uptake for most transition metals is relatively low. Only a fraction of what we ingest gets absorbed. Some, like nickel and vanadium, can also be absorbed via the lungs from contaminated air or smoke.
So low absorption might be protective.
It might reflect our low requirements and the need to prevent toxic buildup. While pathways for some, like iron, are well studied, mechanisms for others are less clear.
Let's take iron as an example. How do we absorb that?
Okay, iron absorption is well characterized. Dietary iron comes as heme iron from meat and non-heme iron, plants and meat. Non-heme iron, usually Fe3+ worn iron, needs to be reduced to F2+ first. This happens at the gut cell surface by an enzyme called duodenal cytochrome B.
Reduced to Fe2+.
Then, the Fe2+ is transported into the gut cell by a protein called divalent metal ion transporter 1, or DMT1. DMT1 is also thought to be the main transporter for Mn2+Un, Ni2+A, and some Cu2+A.
What about copper and molybdenum absorption?
For copper, that same deacetylbenzyme helps reduce dietary Cu+ to C1+. CO1+ is then transported into the cell mainly by a high-affinity transporter called CTR. Interestingly, a high zinc intake can interfere with copper absorption, sometimes causing deficiency.
An interplay there.
Definitely. Molybdenum and vanadium are absorbed mostly as oxyanions molybdate, HMO42, and vanadate, HVO42, likely via a nonspecific anion transporter that also handles phosphate and sulfate.
And finally, cobalt, given its role in B12.
Cobalt is absorbed as part of the vitamin B12 complex, cobalamin, through a dedicated, quite intricate pathway. It involves binding proteins. Haptocorrin in the stomach protects B12 from acid. Then intrinsic factor, secreted in the stomach, binds B12 in the duodenum after haptocorrin releases it. This B12 intrinsic factor complex is then specifically taken up by cubalin receptors on cells in the ileum, the final part of the small intestine, via receptor-mediated endocytosis.
Wow, a very specific process just for B12 and its cobalt.
Yeah.
It really underscores the fundamental importance of these metals.
Absolutely. These diverse, specific mechanisms clearly highlight the active strategies our bodies use to get and handle these essential yet potentially tricky micronutrients.
Well, this has truly been an incredibly deep and comprehensive exploration into the fascinating biochemical roles of transition metals. We've covered a lot from their basic properties like multivalency and Lewis acidity to their specific functions in oxygen transport, energy, gene regulation, detoxification, you name it. We've seen how they act as essential cofactors within intricate organometallic complexes, and also learned about that delicate balance between getting enough and avoiding too much.
Indeed. It's truly remarkable to appreciate just how deeply interconnected these seemingly small players are with the most fundamental metabolic pathways that sustain life. It really shows the incredibly intricate elegant web of biochemistry operating within us.
And it really leaves you thinking about those lesser understood ones like nickel, vanadium, chromium. If we're still figuring out their precise roles, what other undiscovered functions might they hold? What future breakthroughs in health and disease might come from exploring these often overlooked elements?
That's a wonderfully provocative question to ponder. This deep dive has hopefully provided you, the listener, with a solid foundational understanding. We certainly encourage you to delve further into any area that sparked your interest, maybe zinc in gene regulation, iron in oxygen transport, or molybdenum in detoxification. There is always more to learn in this fascinating field.